All right, for this lecture, we are on page 55. We're going to start looking at the cell membrane and the parts of the cell membrane and cell membrane permeability and all of that stuff. So if you don't have your notes out, please get to your binder and find page 55 because there is some things that you're going to want to fill in. So we're going to talk about how the cell membrane is selectively permeable, the role of phospholipids, proteins, and carbohydrates, how water will move in and out of a cell. So we're going to talk about isotonic, hypertonic, hypotonic solutions. And then this electrochemical gradient, I'm going to save that for next time. So I'm not actually going to do that yet today. Once again, kind of like chapter 4, this is just a review of something that you did get in cell bio. There are a few things I will dive a little deeper into, and so you'll see that as we go. Right, so the cell membrane is considered selectively permeable. And so you're going to probably want to write some of this down. I didn't make it to fill in the blankie on this part, so I apologize for that. But you will, there's probably a little bit more writing on this lecture than there has been before. And so you just pause if you need to. But the plasma membrane is selectively permeable. Selective, that means that it lets some things cross while other things it doesn't. The cell membrane is described as a fluid mosaic model. It's described as fluid because there's weak interactions that hold the phospholipids together. And you're going to see there's a picture actually at the bottom there. Really beautiful picture of the fluid mosaic model and everything that is included in that. And so the membrane is held together by weak interactions. So if you can think about phospholipids next to each other, the interactions are between, they're like van der Waals interactions between the fatty acid tails of the phospholipid. What makes the cell membrane mosaic is that you have proteins and carbohydrates kind of just mixed into the phospholipid bilayer, like that picture you can see on the bottom of your paper there. So if you need to pause, go ahead and pause now because I'm going to move on to the next slide. Right, so the early membrane model, sometimes I really don't like showing, sometimes I don't like to show what was the wrong idea, but it's important to see what the history was in trying to determine what the cell membrane actually looked like. So I'm showing you something that's wrong. That's why I said wrong, like really big with a bunch of exclamation points. Davison and Danieli thought that the cell membrane was like a sandwich. So if you look at the picture to the right, the purple would be like the bread. So protein on the outside, I'm like a bread, and then your peanut butter would be in the middle, like would be your phospholipids in the middle. So that's why it's called like a sandwich model. So the proteins are like the bread, the peanut butter's in the middle. So they thought the proteins were on the outside and the phospholipids were on the inside. So phospholipid bilayer in between two protein layers. The problem was that once microscopes were developed and became really, really sophisticated, they found that there was varying chemical composition across the membrane and that there were hydrophobic protein parts. So the protein somehow must be embedded within this hydrophobic part. So this was wrong. It looks like a sandwich. This was kind of just an FYI, so if you didn't finish writing it down, honestly, I don't ask questions about this. I just tell you like, hey, here's what scientists used to think so that we don't take for granted what we know now. I think it's important to see that so that we don't take for granted, and because this is like a stepping stone for what we know now. So even if we discuss, and with evolution, I love to do that. I love to talk about the people who were wrong, or as of today's standards, they're considered wrong. But they were stepping stones to get to what we think is the right answer today. Science is always changing, so who knows? In 50 years, a biology teacher might be talking about this, and they'd be like, God, I can't believe those people thought that. But for now, this is what we believe, and this is what we're going to talk about, and what you're going to answer on an assessment. All right, so you see that beautiful picture at the bottom. I'm still on page 55. There's just a lot of stuff going on in that picture. You see the extracellular matrix that you wrote about on page 54. Remember, on page 54 of your notes, you wrote about the extracellular matrix from the last lecture. You can see the phospholipid bilayer. You can see all the proteins that are embedded within. It's just so beautiful. I love it. I have a beautiful painting on my wall that a student did of that, and I'd always wanted that, and someone finally did it. She did a great job too. All right, so how did they actually determine what the phospholipid bilayer looks like? They did something called a freeze fracture method where they froze it and then they like peeled it across. And so this is how they determined that. You don't need to write that down, but just in case you were wondering. And so they figured out that it's actually a fluid mosaic model that has 2 layers of phospholipids and proteins embedded within, and then you have all your carbohydrates hanging off and stuff like that. What's important to kind of make it, so I'm still on page 55 in case you're wondering where we are, because this like simple picture is basically still represented in this complicated picture. So what you see is proteins embedded within the 2 phospholipid layers, right? What's really important, I want to need you to get from this slide right here, is that if a protein spans the entire cell membrane, it has to have hydrophobic amino acids on the interior and hydrophilic amino acids on the outside. Why? Because the tails of the phospholipid are hydrophobic. And if this interior of this protein, if you're watching my cursor, I'm showing you what I'm talking about. This interior right here wasn't hydrophobic, it would just push that protein out. And we have a word for that. It's called amphipathic. And I am pretty sure I cover that word in just a minute. If I don't, I will go back to it. But amphipathic is what describes a protein that is both hydrophobic and hydrophilic. Oh, it's so beautiful. This is the colored, well, obviously it's the colored version. You don't have the colored version. You know, if you're one of those students, I would totally color this. Anytime you see purple in this class, it means protein. Anytime you see green in this class, it means carbohydrate. And anytime you see yellow, it means lipid. So that's so convenient. I love it that we have it color coded like that. So this just shows you the embedded proteins. This is the inside. The microfilaments are the inside, remember, and then the ECM is the outside. The ECM is, it almost looks like a barbed wire wrapped around a rope, kind of. What I want you to notice is that you have cholesterol embedded within the membrane. You have what are called glycoproteins. So you're going to want to go back to this picture. I know I keep like looking at page 55, but that picture is actually really important. And if you can't see that picture very well, your textbook has it in chapter 5 for you. This exact picture is in chapter 5. I need you to know what a glycoprotein is. A glycoprotein is a carbohydrate hanging off of a protein that's embedded in the membrane. Okay? So then what's a glycolipid? A glycolipid is a chain of carbohydrates. Look how they're hexagons. That's the simple way to represent a sugar. So you have a chain of carbohydrates hanging off of a phospholipid. So you're going to need to know those 2 words, glycolipid, glycoprotein, extracellular matrix, which is made up of, remember what this rope-looking thing is right here? It's collagen. Okay, and then you have your integral proteins and your peripheral proteins. And I am going to go over those words in just a minute, but glycoprotein, glycolipid, that's what you need to get. All right, so the phospholipids are arranged in a bilayer. That means they're 2. Here's the word I was just talking about a minute ago. It's called amphipathic. Amphipathic means that there are both hydrophilic and hydrophobic parts, like significantly hydrophilic and significantly hydrophobic. And so a phospholipid is considered amphipathic because it has the phosphate head, which has the negative charges. Remember, anything with a charge or a partial charge is going to love water. It's going to be hydrophilic. And then it has the hydrophobic tails. And remember, what makes something hydrophobic? Well, it's made up of long chains of hydrocarbons. And when you have hydrocarbons, there's not a big enough difference in electronegativity, so electrons are shared equally. They are nonpolar. They are hydrophobic. All right, all those words go together. If you're struggling with this, you're going to need to look back at chapter 3, where I show you the phospholipid. What the phospholipid bilayer does is it creates a hydrophobic barrier that keeps hydrophilic molecules out. This is convenient because if we didn't have this, whenever we got into like a swimming pool, we would just dissolve. So I'm so glad that we have that lipid bilayer, or we wouldn't be here right now because I'm pretty sure we've all gone swimming or taken a bath or something. All right. A little bit about the membrane and its fluidity. So remember fluid, and normally I would do like this weird motion with my arms, like just fluid, like flowing, not set in one spot, just kind of flowing back and forth, real flexible. I know you've all taken cell bio where you did the bubble membrane lab. Think about that when you think about the fluidity. Think about a bubble and how that behaves. So that fluidity, though, can change depending on temperature. So in low temperatures, so think low temperatures, it's cold. What do things tend to do when they're cold? Well, they tend to solidify and pack together. So think about when you melt butter, it's a liquid, but if you let it sit, if you let it cool down enough, it then becomes a solid and packs together. Well, we are going to be in cold temperatures sometimes. Like lots of plants and animals are going to be in cold temperatures. We don't want our cell membrane to solidify like butter. So our cell membrane is going to do something that adapts to cold temperatures, and it's going to create kinks in the tails. And so what creates a bend in a fatty acid chain? It is a double bond between carbons. So in low temperatures, the phospholipids of the cell membrane are going to form double bonds between carbons of the fatty acid tail, and that creates bends. And when they're kinked like this, look at where my cursor is. When they're bent and kinked like this, it creates a space so that they don't pack together and form a solid. All right? Hopefully everyone got that. Cholesterol also helps in low temperatures. So down here, look at my cursor. Anytime you see 4 fused rings together, and especially if it's yellow, it's going to be cholesterol. It's a lipid. That is not a carbohydrate because they are sharing sides. It's fused rings sharing sides. So if you look, I want to show you the difference. Do you see this carbohydrate? They're not sharing sides. The points are meeting, but they're not sharing sides. But if you look down here at this cholesterol, okay, those rings are fused together. Those are hydrocarbon rings. So I know that's kind of tough sometimes for students. When they see rings, they just want it to be a carbohydrate so bad. But for cholesterol, it's not. It's a lipid. So cholesterol is going to help in low temperatures because it's going to help resist changes, and it's going to hinder close packing at low temperatures. Cholesterol is also going to help with high temperatures. So you can go to the opposite extreme where it's too hot and the cell membrane becomes like kind of melty. So we don't want it to become melty. And so cholesterol is going to be there to prevent that also. So if I asked you, how does the cell membrane adapt to cold temperatures? You would say double bonds between carbons, also known as unsaturated fats, and cholesterol. And then if I asked you, what is the adaptation for the cell membrane when there's high temperatures? You would say cholesterol. So cholesterol works for both. Okay, so if I asked you if you had bacteria in a hot spring, what are you going to see a lot of in their cell membrane? You would say cholesterol. If you have winter wheat and you don't want it to freeze and you don't want the cell membrane to pack together, what are you going to see a lot of? Unsaturated tails and cholesterol. All right, so hopefully you got that. All right, we are down to, so hopefully you got all that down and answered all those questions. If you need to go back, you want to go back. It's important notes to take. And y'all, actually the lectures are like the most important part of the class, which is why I pre-record them so that you have them always and you can rewind. So make sure that I think that needs to be a focus, is make sure that you're watching the lectures and taking the notes, because that is like the class. That's the information. And then class time is used to review it and do like extension activities and stuff like that. So I just want to make sure I said that. I think everyone already knew that, but I just wanted to make sure I pointed it out again. Integral proteins and peripheral proteins. Integral proteins are embedded in the membrane. Don't even write that down. I don't care about you knowing about freeze fracture. Transmembrane with hydrophilic heads, tails, and hydrophobic middles. They're amphipathic. So integral proteins are embedded and they're also amphipathic. Peripheral proteins are extracellular or the cytoplasmic side, but they're like not embedded. They're either held in place by the cytoskeleton or the ECM. So what kind of peripheral protein would be held by the cytoskeleton? Well, that's on the inside of the cell, so that would be the cytoplasmic side. What kind of peripheral protein is going to be held in place by the ECM? Well, that would be an extracellular peripheral protein because that's on the outside of the cell. All right. Their job is to provide stronger framework. So pause if you need to pause because I'm moving on. All right, here's another picture. I'm showing another picture here. You have your extracellular matrix, your glycoprotein, more carbohydrate, that's carbohydrate, glycolipid again. There's your cholesterol right there. There's your integral protein and your peripheral protein. Your integral protein, remember, is amphipathic and it spans the entire membrane. Your peripheral protein's kind of hanging off the side. This is an this is a cytoplasmic peripheral protein. It's held together by the cytoskeleton. It's held there by the cytoskeleton. I don't see in this picture, I don't necessarily really see any extracellular peripheral proteins. But if you did, it would be kind of like hanging off right here. Maybe this one right here. We can kind of we can consider that one right there actually to be a, yeah, we consider that one right there to be an extracellular protein. So you can see where my cursor is. All right, so just so you know, there's the picture. This is just telling you transmembrane protein and integral protein. We are just considering those to be the same thing, and they're amphipathic, once again. This is just giving you a little bit more detail on what that looks like. I wouldn't get caught up in this picture. I just want you to know that integral proteins are also known as transmembrane proteins, and they are amphipathic. They must have hydrophobic interior, hydrophilic outside. That way they can stay embedded. Don't worry about N terminus, alpha helix, C terminus. That really isn't something that I see as a big part of this class. That, as far as I know, I'm just trying to cut out as much extra information as I possibly can, just because of our limitations this year. And I just don't think those words are necessary. But you do need to know amphipathic, and you know what amphipathic means. And if you don't know what it means, just look at this picture, and that'll tell you. Ooh, okay. Some jobs of membrane, integral, and transmembrane proteins. They have jobs. Okay. Transport, enzyme activities, signal transduction, cell-to-cell recognition, intercellular joining, and attachment to the ECM. Our goal for this chapter is A, transport. Our goal for one of our upcoming chapters is then going to be enzymes and then signal transduction. So I would say A, B, and C are the ones that we're going to talk about the most in this class. So pause if you still need to write those down, because it looks like on page 56 you need to list those, because I'm about to click. All right. The job of the carbohydrates, like what are the carbohydrates? What are the carbohydrates doing hanging off of the cell membrane like that? Well, they're there for cell-to-cell recognition, and especially in developing organisms. You have your glycolipids and your glycoproteins. Those are your examples. And then an example of why carbohydrates on the cell membrane are important would be blood transfusions. I talk a lot more about blood type, and if you had me this summer, you got the opportunity to do blood typing. If you didn't do it, or if you didn't have me this summer, we do blood typing kits in my class. I'm not sending them home this time. We're going to actually do them in class, and I actually perform it for you. I think some students are scared to do it to themselves. But the carbohydrate hanging off of your red blood cells is actually what determines what your blood type is. And if you are transfused with a blood type that doesn't have a carbohydrate that is recognized, then that's when you have problems. And so that's why they talk about blood compatibilities and transfusion compatibilities and stuff. All right, I'm about to click, so you can pause if you need to. All right, synthesis and sidedness of the membrane. Oh, so what do carbohydrates look like in the drawing? They just look like a bunch of hexagons chained together. Synthesis and sidedness of the membrane. Honestly, I would just skip this part. Yeah, we're just going to skip this part. Synthesis and sidedness. I'm going to address it during the Zoom call, so let's just skip this for now. I mean, honestly, it's just the ER creates vesicles that fuse with the Golgi, and then the Golgi creates vesicles that fuse with the membrane, and then the membrane releases content. I think what this is really just saying is that it's a continuation of membrane being used on the inside and the outside, because it can do the opposite. Things can come into the cell and then use vesicles to move around. So I would just say membrane is used as vesicles for movement through the cell and out of the cell, things out of the cell. Let's talk about how things can go across the cell membrane. Okay, so we're going to talk about transport. I do have to go through page 58, so I'm sorry that this is going to be over 20 minutes. The membrane is selectively permeable, so what can freely pass through the membrane with no problems are small molecules, polar or nonpolar, hydrocarbons, and hydrophobic molecules. You're going to want to memorize that. You're going to want to make a flashcard. Small molecules, polar or nonpolar. Water is actually considered a small molecule that can pass through the cell membrane. Hydrocarbons, like cholesterol, testosterone, estrogen, and other hydrophobic molecules, carbon dioxide and oxygen. So memorize that, please. The hydrophobic core prevents the passage of ions, which are charged, and large polar molecules like sugar. Yes, like sugar. So I'm going to click. Go ahead and pause if you need to. All right, passive transport, let's talk about this. Does not require any energy. So we're talking about diffusion across the membrane. So it does not require any energy, no ATP needed. Diffusion occurs down a concentration gradient from high to low concentration. And our examples of things that can diffuse freely across the cell membrane are hydrocarbons, carbon dioxide, oxygen, and water. Okay, so concentration gradient from high to low, and then what can freely diffuse across the cell membrane: hydrocarbons, carbon dioxide, oxygen, and water. Once again, I would have those memorized. And so this is just showing you what diffusion is. It's the goal of diffusion is to create equilibrium, equal amount. So if you look at the dye in the water, why does dye spread through water? Basically, it's to achieve equilibrium. Okay, I'm going to click. All right. And so this is what diffusion is. Diffusion is just the movement of a molecule from high to low concentration. So all I would write is just movement from high to low concentration to establish equilibrium. All right, so under diffusion on page 57, movement of a molecule from high to low concentration in order to establish equilibrium. Osmosis is the diffusion of water. So the movement of water in order to achieve equilibrium. And so what is used in order to demonstrate osmosis is this thing called a U-tube. A U-tube. So if you look at this tube right here, you see that there is a selectively permeable membrane. Water can pass, but these sugar molecules can't. Naturally, the sugar would go to the left, right, to make it equal on both sides, but it can't. So water is going to move in order to establish equilibrium. And the equilibrium that is established here is equal concentration of sugar on both sides. Okay. Let's discuss hyper, hypo, and iso. On the bottom of page 57, please change hypertonic to hypotonic. Change it to hypotonic on the bottom of page 57. This should all look familiar to you. This picture right here should, well, if you took me in for cell bio, you definitely saw this picture. You got this like exact lecture actually. So I tried to set you up well for this. All right, so on bottom page 57, you see it says hypertonic. You changed it to hypotonic. So hypotonic solution, or hypotonic environment, the concentration of solute is going to be greater inside the cell. Therefore, water moves in, an animal cell will lyse, and a plant cell is turgid. Okay? I'll say it one more time. In a hypotonic environment, the concentration of solute inside the cell is greater than outside. So water moves in. Animal cells will lyse or explode. Plant cells will become turgid or normal. So animal cells are sad in a hypotonic environment. Plant cells are happy. Yay, I can stand straight up. So why is it that a plant cell isn't going to explode? Because it has a cell wall to push back on the water expanding, and therefore the plant becomes turgid and very happy and stands straight up and gets sunlight and does photosynthesis. Yay. This is what would happen if you went to the hospital and you had an IV where they infused plain distilled water into your veins. That's why they can't do that. That's why when you look up and you look at your IV bag, it's going to say 0.9% NaCl because it has to have a solute that matches so that your cells don't explode. So speaking of a solute that matches, that would be an isotonic solution. Isotonic solution, the concentration of solute on the inside of the cell is equal to the concentration of solute on the outside of the cell. That makes animal cells very happy. There's no net movement of water. Water just moves in and out at equal rates. That makes plant cells really sad and they start to droop over and become flaccid. Aw. Okay. So rewind out if you didn't get it. This is very important to write down. I'm sorry I didn't make it fill in the blanky enough. All right, hypertonic environment. The way I remember hypertonic is if people are hyper and they move a lot, they'll lose weight, I guess. Hypertonic means that the concentration of solute on the outside of the cell is greater than that of the inside of the cell. Okay? Therefore, water moves out. Animal cells become shriveled. Plant cells become plasmolized. Plasmolized. You're going to need to know these words. Definitely make some flashcards for these. We are going to do our lab setup, and then we're going to work on water potential next week. I will talk to you guys later.